Okay, thanks very much for the opportunity to present today. Sernova is a cell therapy regenerative medicine company, and I'm going to talk about our diabetes programs that are ongoing and a number of our other applications. Please be aware of the forward-looking statements. So a couple of company highlights. First of all, we have what we call the cell pouch system, and this comprises a small medical device that is placed under the skin that forms an organ-like environment into which we put immune-protected therapeutic cells. And we believe these cells need to be contained in a device to be able to function properly so that you have control over what cells you're putting in. And if there's issues, the idea is that the device can be removed as appropriately. And also, we can understand a lot about cell survival as well as the vascularization and release of materials or proteins, et cetera, from the device. So our lead application is type 1 diabetes, but we're also working in two other areas, which is hemophilia A, which is an ex vivo gene therapy approach, and also thyroid disease. The company is well funded, which is something unusual in this day and age. We've got over $50 million in the bank, which provides us with at least a 2-year runway to run all of our programs. And we are on the Toronto Stock Exchange, and our anticipation is to move to NASDAQ at the right time when the market conditions improve. So if we get a little bit more into our technology platform, there are 3 parts of this. The first one is the cell pouch. And what's really important about medical device for therapeutic cells is that you've got to create an environment for those cells to be able to survive. And so our device is shaped into a series of small cylindrical chambers, and the polymers itself provides a very, very porous environment so that when we place this device deep under the skin, it rapidly fills in with highly vascularized tissue around small removable plugs. And the tissue grows to the circumference of the plug. And then after about 3 weeks, we can remove those plugs. And then we have a void space for placing the therapeutic cells. And those therapeutic cells will then engraft, and they're sitting in a proper tissue matrix, and then they'll be able to produce their product as required. With respect to the therapeutic cells, our approach has been to de-risk the technology to work with human donor cells. So our first clinical trial with type 1 diabetes is using human donor islets, but that obviously has a small supply of cells that is for patients going forward. And we have done a deal with Evotech to provide an unlimited supply of iPSC fully developed islet technology going forward. With respect to the immune protection, there are multiple opportunities that are being developed right now in the field, and we've chosen to work with a polymer approach, which is called conformal coating. And this is a cross-link polymer that surrounds the cell, very similar to a shrink wrap approach, and it's been shown to provide normal kinetics of insulin release and glucose at this point in time. But of course, there are other ideas that we're also working on in the background. So very quickly, to talk a little bit about our technology, we place the device deep, deep under the skin at the level of the muscle. After about 3 weeks, it has these fully formed vascular sites to be able to put the therapeutic cells in, and then those cells will engraft to start the function. Again, I won't go over this slide, but we're working in 3 different areas, and you can see that we have a number of products in each area, starting off with the human donor cells, and then moving through with stem cell-derived technologies to be able to expand our patient population in terms of the treatment. So with respect to our University of Chicago ongoing phase 2 trial right now for type 1 diabetes, we are working with patients that have long-term type 1 diabetes, a history of severe hypoglycemic events, and no ability to produce insulin. And this is measured by a biomarker called C-peptide. So the important thing for us is that when we place the device into the body and we place the cells in it, we're working on understanding not only the dose of cells required to get efficacy, but also the density of the cells that need to be in the device. Because as you probably understand with islets, they need to have space to be able to read the natural blood sugar levels and not turn each other off. So primary endpoint here is safety. Secondary endpoint are survival of the endocrine cells in the device itself, because multiple competitors in this field have failed due to fibrosis. So we want to ensure that those cells are surviving. Secondly, we're looking at a proportion of patients with a reduction in severe hypoglycemic events, and then also we're observing hemoglobin A1C to look for a reduction there. And finally, we have over 20 additional endpoint analyses that we're doing in this study. So these results, partial results, were presented at the American Diabetes Association meeting this summer. And what we found, and we have continued to find this, is that the implantation of the device is safe. And patients have had devices in their bodies up to 3 years with no safety issues whatsoever. And importantly, we've treated 6 patients. The first 3 patients have achieved insulin independence, and the longest patient is at approximately 2.5 years now. So we're showing that the technology works. We've had several patients that did not take the optimal immunosuppressive drugs, and those patients are now have been corrected, and those patients are now moving on with additional islet transplants in the devices, et cetera. Importantly, as you can see, we have HbA1c in our 3 patients in a range of 5, which is normal for these patients. And additionally, they're getting no hypoglycemic unaware events, and they're living their lives normally. So if you can think about these patients that have been taking insulin for 20 years and now they're completely off insulin, this is what we would call a functional cure. So with respect to our histology that we're seeing inside the devices, we have small sentinel devices that are in the patients that come out at the 90-day time point after each transplant. And we're consistently showing abundant blood vessels, surviving islets, and the ability to produce all of the hormones that are required to control type 1 diabetes. So we always talk about the science, but what really matters here is what the patients are saying. So this first patient that is now off insulin for 2.5 years, to give you a little bit of an idea, she's taken over 20,000 injections of insulin prior to working on the trial with us, and had type 1 diabetes for over almost 50 years. She's been on insulin pumps, she's been to the doctors many times, and she's now been insulin independent for 2.5 years right now on the device with the human donor islets. So when we look at the human donor islet program, this is really establishing a flag in the area for us in this field and getting a potential of having a product approved as a first step. But our next step moving forward is to be able to treat all patients with type 1 diabetes using an iPSC-derived technology. And we announced just in May the deal that we've done with Evotec. And in this process, we actually spent a number of years working with Evotec in the background, working on their cells in combination with our cell pouch, and we got beautiful results. And we then parted ways for a short period of time. Evotec went out and tested every other device that they could possibly find. They came back to us last August and said, we need to do a deal. Your device is the only one that we find is working. So in this regard, we did a deal with Evotec whereby we're sharing in the development of the cells in the cell pouch up to the IND phase in terms of costs, and then after that, Sernova will take over the clinical development of the product. And what is really important here is that Evotec is in charge of manufacturing the cell product for us all the way to commercialization. In that regard, they have purchased an iPSC GMP facility in Italy. There's been a lot of discussions around manufacturing of cell therapies, and we have that completely sewn up. And I'll talk about that in just a little bit more. So with respect to how this is going to work in terms of patients going forward, what we're anticipating is that our cell pouch and surgical kit would go to any hospital in the world, and the endocrinologist would then call the manufacturing facility, and those cells would then be shipped live to the site where they would go directly into the cell pouch. So we have this already figured out on how the patients are going to be able to receive this product. And what's really important here is that it eliminates the need for us to go to the small number of islet transplantation centers, and we're anticipating that we'll be able to go to virtually any hospital in the world that has a general surgeon that can slip the pouches in and an endocrinologist that can be able to work with the patients. So when we're looking at the cells from Evotec, one of the really important parts in terms of manufacturing is that these cells are highly controlled. So the results that we're seeing in our islet program, if you imagine islets have to be taken from a donor pancreas and then they go through a whole release criteria process and then go into the pouches. With these iPSC-generated cells, it is going to be much more like a pharmaceutical product, and they are tightly controlled at every part of the differentiation protocol with release criteria going forward. And what is really important here is that the cost of the manufacturing process is under control too, unlike other companies that are working in this field. And in our process, we get the lots up to the point where we can cryopreserve them. And then once the physician orders the product, we can then take those islets and then run them through the last part of the differentiation protocol, and then they are shipped live. What's also important about these cells is that they're three-dimensional, and they have been designed over a 6-year period to be able to have the same components as normal human islets do. So the production of C-peptide is normal, the production of insulin, and the other hormones. Preclinical studies have been done for up to a year, and we're right now in the process of combining our technologies, scaling up the product, doing tech transfer, and getting ready for clinical trials that will be starting in 2024. So as we're going forward with the diabetes program, our plan is to move forward with our phase 2 clinical study. And what we have found is that we want to increase the number of cells that are going into the devices. And so we've manufactured slightly larger devices that will provide enough cells for the patients to not only get efficacy, but also to be able to endure any kind of metabolic stress, such as COVID. So our first patient, for instance, did get COVID, and we were a little bit worried about what would happen, but this individual was able to sail through and is still insulin independent. So it's really teaching us a lot about what kind of dosing we need to do in all patients as we're going forward. Importantly, the reason for continuing this on for the potential of doing a phase 3 study is so that we can get a product approved while we're working on our beta cells from Evotec going forward. So our plan is to have a number of products in sequence that are going to be approved for patients to be able to use. So with respect to our conformal coating technology, this is something that's been worked on for over 12 years at the University of Miami and optimized. So this is a cross-link polymer coating that is placed around each islet or each cell, whether it's an iPSC cell or an islet itself. And it's a hydrogel type coating. And the idea is that it doesn't take up any room within the cell pouch chamber. So it's an ideal type approach to be able to use for our technology. And Sernova has licensed and has worldwide exclusive rights to this technology. So this just gives you a little bit of an idea of what the cells look like. In the center, you can see the white circles around the conformal coated cells. These are individually coated. And when we place these into the cell pouch, what we've been finding is that they're producing the insulin and glucagon that they're supposed to. And we've already done a number of studies in gold standard animal models for type 1 diabetes and been successful. So look forward to this moving into the clinic. And we think that this is going to be our first step in terms of having a local immune protection technology for all of our cell therapy approaches that can be used to help patients eliminate the need for the anti-rejection medications. So just very briefly, we believe that we're the first company to work with patients that have benign thyroid disease, and we're working with patients that are having their thyroid gland removed. And what we've been able to find is that we can take the healthy portion of the thyroid gland and replace that back into the device. And we've done successful preclinical studies to show that we can bring back the hormone, not only the hormone levels, but also the feedback loop between the thyroid and the brain itself. So we're looking at taking this into the clinic in 2023 and are working on the regulatory documentation currently right now. So again, this paper has been published around the results from this. We are also working on an exciting program for hemophilia A and other potential rare diseases, and this is an X-gene therapy program. So here we've worked for 4 years on a product whereby we're able to take a sample of the patient's blood and then insert the gene for factor VIII into the endothelial cells, place it into the device, and we've been able to show that we can get functional levels of factor VIII into preclinical models. And again, this has also been published. So We have a number of near-term catalysts going forward, and I won't go through these because we're pretty much out of time, but I just want to thank you for the opportunity to talk today.